Chapter 13, Section 3, Phosphoryl group transfers and ATP. Our metabolism is composed, for all intents and purposes, two sides of a coin. Heads, catabolic reactions, tails, anabolic reactions. Catabolic takes the big things, the fatty acids, the polypeptides, the long strands of nucleotides, and breaks them down into small things that can then be fed into, our cellular respiration, ours being living cells, whether it's the citric acid cycle, which can then be used, those excited electrons can then be used to power oxidative phosphorylation and the making of ATP. The anabolic portion, the tails of our coin, takes small things and builds them up. Takes small things and pushes through reactions. So it will give us products that can then be used to make things, the subunits of, what will become the amino acids. Takes the amino acids, links them together, becomes the polypeptides as an example. The problem is the anabolic takes energy. Catabolic yields energy. But we have to have a way to link the two sides of the coin together. That's where ATP comes into play. All that energy from the catabolic is going to be captured, transferred to proteins and protein complexes in the mitochondria, which will then, the mitochondria, powerhouse of the cell, will make ATP, which will then power anabolic reactions. What's interesting is pretty much all cellular organisms of this planet have evolved to focus on ATP. ATP is this linker molecule that links, takes the energy from catabolic reactions, which can then be used in anabolic reactions or other metabolic requirements throughout the cell. And it has to do, you start with the adenosine nucleoside. Remember, the nucleoside is the sugar, the nitrogenous rings. And then onto that nucleoside, you add 1, 2, 3 inorganic phosphate groups, PO4. Alpha, beta, gamma. Start numbering from the nucleoside out. Energy, energy, energy in those bonds. Well, we want to release that gamma inorganic phosphate, that third phosphate group. What you're going to have is a nucleophilic attack on the phosphate, which is going to cause shifting of our electron pairs. So we talked about in the last section, the lecture of the last section, this incoming oxygen, here it's from water, it could be from a hydroxyl group if we're talking an enzyme. Oxygen is doing nucleophile attack. Phosphate group is going to release, for all intents and purposes, release the electron that's holding it to this oxygen here. This oxygen is going to stay with that phosphate group. As we see down here, that phosphate group is now going to be heavy in electrons, so it's going to have a polar negative charge. But this oxygen here is going to take the place. See? Red, red, blue, blue. But what's interesting is the double bond we see here with this upper oxygen. This isn't a stable, it will always be in this, because remember we talked about that last section, the lecture in the last section. That double bond there will move between these four oxygens. The hydrogen kind of gets left out. It's still there along for the ride, but it's hydrogen. It's a proton just sitting out there. So that's why you get this resonance. You see here, at any given instant, all of these oxygens will take on a negative-ish charge. Any oxygen that's not double bond or has that hydrogen, that proton there balancing it, 1, 2, 2 negative charges. But here, it bounces around, it resonates, that double bond. So at any given instant, where's that double bond? Where's that double bond? It moves around. So what we would think of as, HPO should be 1, 2 minus O4 becomes 3 because, well, that hydrogen just kind of, that proton's just hanging there, and then the rest of them will share that double bond, and it's instantly moving around, so it now becomes a pseudo 3 minus charge. That's just the inorganic phosphate. The remaining A diphosphate, remember di means 2 phosphate groups, it now takes on this ionization, bounces back and forth between these two forms, where this hydroxyl will lose the hydrogen or gain a hydrogen back. So again, at any given instant, it could be ADP 2 minus or ADP 1, 2, 3 minus, depending upon the solution it's in and what's happening in the organelle that happens to be in, if it's an eukaryotic cell. And when we look at this ATP, ADP, and inorganic phosphate concentrations, or even if we take in the phosphocreatine, which is used by some cells as a quick means of taking a diphosphate and turning it back into a triphosphate. A lot of times you're going to see this in vertebrate muscle cells that don't have the time to wait for that whole oxidative phosphorylation in the mitochondria to renew ATP. They need it now. Boom, 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 because of fast twitching. So you'll notice certain cells need more than others. Hepatocyte, myocyte. These are going to be very metabolically active. Lots of things going on. Neurons, while metabolically active, they don't do a lot of repair. They don't do a lot of, making of new things, of having to go through mitosis and things like that. Whereas a hepatocyte, a liver cell, yeah, it's going to be a lot going on there. Erythrocytes, human erythrocytes, are red blood cells. Some metabolic activity, yeah, so going to need some ATP, but not a lot because it's got a short shelf life for red blood cells. Here today, gone tomorrow. But you'll notice the higher the ATP concentration, the lower the ADP, the lower the AMP. A triphosphate, a diphosphate, a monophosphate. So what we see is that the cells have a tendency to push AMP to ADP, ADP to ATP. They keep around some levels of AMP. Turns out AMP does have roles in the cell beyond being made into a diphosphate, so it can become a triphosphate. When you're doing some studies, signaling studies, or you're studying metabolism in cells, and you're looking at what's happening, reactions, and you're trying to mimic in vitro what happens in vivo, you'll see a lot of the buffers that you'll have to use that not only are buffering against changes in pH, but are also including in certain salts, certain minerals. Magnesium is one of them. Turns out magnesium interacts with, sorry about that, I'm playing with coins as I'm talking. Magnesium interacts with a triphosphate. Magnesium 2+, it's not bonding, there's no electrons shared because the oxygen is busy already. But it has an interaction with these two oxygens, forms this pseudo cyclic ring. Same here. What this does is that it stabilizes this overall structure so that it doesn't inadvertently start interacting with other molecules, or the things that have charged or are looking to strip off charges. We know delta G is a measure of how much free energy. Delta prime, delta G prime naught is, the free energy under given standard conditions. Certain pressure, certain temperature, certain concentration, blah, 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 blah. Well, we also have a delta GP, which is the phosphorylation potential. This deals with a triphosphate. How much energy is being brought into a system, how much energy is being taken out of a system due to metabolic reactions surrounding a triphosphate. When you look at it, the energy released by ATP hydrolysis is greater than the standard free energy change delta G prime 0. Because remember, ATP, when we measure it, we calculate it negative 7.3 kilocalories per mole, negative 30 kilojoules per mole. But we actually, when we look at it, we actually see more going on there. So biochemists had to come up with a mechanism for studying this, for describing this. Evolution, as I said at the beginning of this lecture, has selected for ATP. Okay? Don't know why. Don't know what it is about ATP, a triphosphate in particular. But what we know is that when ATP levels drop, fuel decreases, what's there loses potency, cells start to slow down, cells become sluggish because metabolism is affected. When we go and we look at, you look at here, table 13.6 from the textbook. My head's covering some of the numbers, so you'll have to look in the handouts or in the textbook. You'll see that when we step back and we look at certain biomolecules, phosphoenol pyruvates, the second to the last step of glycolysis, when you end up with pyruvate. I mean, look how much energy is there. Negative 61.9 kilojoules per mole, negative 14.8 kilocalories per mole. Here's our friend, ATP. So breaking that down to a diphosphate, negative 30.5 kilojoules, negative 7.3 kilocalories. This, isn't the highest, isn't the lowest. Many of the things are metabolism, and when I say are, I mean living organisms. Because you go through and you think back to your intracellular molecular cell biology courses, many of these, glucose 3-phosphate, fructose 6-phosphate, glucose 6-phosphate, these are going to be molecules that are involved in glycolysis. We're taking the sugars, we're going after carbon-carbon energy, we're going after some of those excited electrons. So we see there is a lot of energy that needs to be bled out, taken out under specific conditions to minimize second law of thermodynamics, going to be captured, transferred to the mitochondria, so more of this can be made, a triphosphate. And you look at this. I pointed out the phosphoenolpyruvate because PEP. Got to remember, scientists, we hate writing out phosphoenolpyruvate all the time. We have these big letter words, so we'll use acronyms. Acronyms are accepted. PEP. 1, 2, 3. 3 carbons. Carboxyl. 2 hydrogens. Middle carbon, C2. Oxygen, phosphate group. This oxygen will become a carbonyl, but this phosphate group has to go. Hydrolysis, remember, to remove phosphate, going to have some form of a hydrolysis reaction, so we need to bring in water. So we bring in water, this oxygen will, nucleophilic attack on that phosphate, which is acting on the electrophile, so strips it off, have its 4 oxygens, it's released as an organic phosphate, and then you end up with pyruvate. The pyruvate, though, goes through what they refer to as a tautomerization. There's 2 forms of it that it will sit there and basically go back and forth between. Now, I'm assuming the artist who drew this is showing this one long arrow, this short one. Pyruvate, enol form to keto form. Boom, boom, boom. Seems to be a way it wants to go. Yes, it will bounce back, go the reverse, but not quite as easily as it wants to change. Swap out this hydroxyl, have it become a carbonyl, and this becomes a methyl, or CH3. PEP3 to pyruvate. Look how much energy is released there. That energy is coming here in this phosphate group, which is being pulled off, and then will be enzymatically attached, substrate level phosphorylation, by an enzyme that's acting as a phosphorylase. Remember, talked last section. Phosphorylases add inorganic phosphate to their substrate. Here the substrate is going to be a diphosphate. Substrate level phosphorylation gives you an ATP that looks normal, but it is slow and cumbersome compared to the making of ATP like we're going to see in oxidative phosphorylation in a few chapters from now. Excuse me. When you look at many other reactions that are going to be occurring in glycolysis, occurring in the citric acid cycle, some of the products of this hydrolysis of the phosphate group are going to be of such a structure that it will have this, tautomerization, this ionization that can occur. Here you take 1,3-bisphosphoglycerate. Okay, carbon 1, carbon 3, both have the phosphate groups. Hydrolysis reactions pulling that up, inorganic phosphate off, which will go to an A diphosphate using a phosphorylase. Okay, boom, we get an ATP there, leaving a carboxyl group. Thing is, though, this carboxyl group wants to ionize. That hydrogen is going to be released, and you'll have carbon, double bond oxygen, single bond oxygen. And just like we looked at a few slides ago with the inorganic phosphate group, that double bond orientation is not going to remain stable. Okay, it's going to change. So you have this resonance. Any given instant, oxygen on the left has the double bond. Any other given instant, it's the right, back to the left, back to the right, boom, 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 bouncing back and forth, and they call this a resonance. So that double bond bouncing back and forth actually stabilizes. We still, when we draw it out and we discuss it as a carboxyl group. But in the reality, that hydrogen's being released and you have this occurring in the aqueous solution. Is this reversible? Yep, because you got to remember, all aqueous solutions are going to have free hydroxyls, free hydrogens, just because, the water's breaking down on its own. So there's always going to be a hydrogen there at some point in time that will come back and then be released again, come back, be released, and this stabilization will continue to occur. Phosphocreatine, remember, mentioned this is used by certain cells, specifically muscle cells, use the highest rate of it. Muscle cells of vertebrates specifically. So you have this phosphocreatine that has this phosphate group sticking out there. It's a little bit different than the quote-unquote norm phosphate groups that we've seen before. There's no P, oxygen, N. No phosphate, oxygen, N is the phosphates directly to the nitrogen here. So it's going to be a little different. Again, hydrolysis reaction, this oxygen will replace where the nitrogen is, that electron pair there. So you have the release there. These hydrogen, one of these hydrogens will fill in there. So we have the electrophilic attack of this, or nucleophilic attack of this oxygen to that phosphate, which will act as an electrophile and will take the electron, and this gets released, which will then be ultimately added to an A diphosphate to make A triphosphate using substrate level phosphorylation, and the ATP levels of a cell that's going under stress, under hard work, and the mitochondria can't seem to keep up. This will give that last little boost of energy that's needed to keep things going, keep that muscle twitch going. Phosphocreatine to creatine, or if you're British, creatinine. Nitrogen, carbon, nitrogen, methyl, carbon, carbon, boom. What we see here, again, is a resonance effect. Single bond, double bond. Who's got the double bond at any given instant? Will change so that you will get this resonance. Notice, still NH2 here, but this is N plus H2, and this is just NH2. Well, at any given instant, you end up with both of them being positive-ish. Not true positive like you see here, but positive-ish, because now I have it, now I don't. Now I have it, now I don't. Okay? A lot of energy right there. Now, students have asked, okay, to get all this energy going from phosphocreatine to creatine, and, some of it's going right there, but not all of it. Where's the rest of it go? And I always remind them, 98.6 degrees Fahrenheit, 37 degrees Celsius. And we see a lot of this also, this resonance, thioesters, okay? Carbonyl, sulfur, CoA, acetyl-CoA. Acetyl-CoA is a 2-carbon thing, carbonyl, sulfur, hydrolysis to release the CoA, and you end up with a carboxyl. A carboxyl group that's in an aqueous solution. Remember aqueous solutions. That means that this hydrogen, as we see right there, gets released. As it gets released into the water solution, the water, we again get this resonance. Who's got the double bond? Who has it now? Who doesn't have it? And here, instead of being the positive, like we saw with the nitrogens on the last slide, here we see that with these thioesters, again, we go back to what we saw a few slides ago, where it's a negative-ish. Boom, 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 back and forth. Whoever has the double bond in that given instant is neutral. Who has the single bond, because this hydrogen here has been released, now is the negative. Excuse me. Now, it turns out there is a difference between thioesters and oxygen esters. Who's being released? Who's not being released? Turns out these thioesters have a higher energy, higher innate energy levels than the oxygen. Goes along with more needed for stabilization of what is occurring here. So just because you see an ester that's being hydrolyzed and you see it going through doing this resonance, not all of these resonance stabilizations are going to be the same. And the energy that's coming from this ATP isn't just because a phosphate group is being, it's not just because there's a hydrolysis occurring. Ooh, look, we broke a bond, energy's released, boom. No, the energy's coming in because a functional group is being transferred. Yes, I use the very simplistic analogy of a light switch. Okay, adding on this functional group does change things. Whether it's being added directly to an intermediate, so you can go from glutamate to glutamine, or if the functional group, the inorganic phosphate's being transferred from a triphosphate to an enzyme to activate it. It's changing the shape, change the function. So when we look at this reaction of taking glutamate, swapping out this oxygen here of this carboxyl and adding in an amine group, going from glutamate to glutamine. You'll see it in many books. It's glutamate plus ammonia, some energy from ATP, and you have glutamine. When in actuality, it's a multi-step process. Hydrolysis of ATP attacks this carbon here, puts in inorganic phosphate. The ammonia attacks the same carbon again, displaces this inorganic phosphate, releasing it, and now you have an amine group here attached to the carbon chain, giving you glutamine. And when we look at the chemical structure of many of these compounds we're going to see throughout cellular respiration, there is a spectrum on how much energy is imbued within the compounds. Again, I've said it before. The more negative a delta G is, whether it's the delta G or the delta G prime naught, the more negative is, the more energy is there. I mean, phospholynyl-pyruvate. That phosphate group is going to be transferred to ATP, giving it a lot of energy. Still a lot of energy just in the pyruvate itself, which is going to be transferred over to the mitochondria for citric acid cycle. Phosphocreatine, remember that phosphate group we just talked a few slides ago, gets added to A diphosphate to give energy to A triphosphate. Just because you're cutting off the phosphate group here doesn't mean you lose all the energy. Phospholynyl-pyruvate becomes pyruvate. There's still a lot of energy in pyruvate. So it will drop down, but it doesn't completely go away. And it's not just the inorganic phosphate itself. The inorganic phosphate doesn't really have a lot of energy in it. It's how it's being transferred, what's being transferred, what it's being transferred to that allows other things to do something. That's where the energy is. So you take those high energy ones from the last slide, PEP. PEP to pyruvate. Remember, I told you that's the last step of glycolysis. You go through hydrolysis reaction, remove that phosphate group, and now it just becomes pyruvate. Negative 61.9 kilojoules per mole is the delta G. You take a diphosphate and that phosphate that was released here, add them together to form ATP, substrate level phosphorylation. That's a very high delta G. That takes a lot of energy. Thing is, told you, the first lecture of this chapter, the delta G's in a multi-step reaction like this are all additive. You go through, you cross stuff out. PEP plus water yields pyruvate plus inorganic phosphate. ADP plus inorganic phosphate yields ATP and water. Waters cancel, inorganic phosphates cancel, so you end up with phosphoenolpyruvate plus adenosine diphosphate yields pyruvate and adenosine triphosphate with a delta G of negative 31.4 for this complete reaction. Negative 61.9, positive 30.5 yields negative 31.4. When you look at them, remember alpha, beta, gamma. Each of these can be attacked. The majority of the reactions we'll talk about, it's going to be this gamma phosphate group. It's going to be the one that's released. But there are certain reactions that beta will be attacked. So you have the two inorganic phosphates being released at the same time. Very, very rarely. That's especially if you want to have resulting AMP. Because remember, I told you a few slides ago, AMP has uses in the cell beyond just being the base molecule for A triphosphate. So there you go. Cut off beta gamma, you end up with AMP. AMP will then be used to power certain things. No, not power. It'll be used as an intermediary in certain reactions. It's part of certain reactions. Cyclic AMP is one of our secondary signal molecules. Cut off beta and gamma, you end up with AMP. Adenylyl cyclase will then take that and, make it cyclic, and you have cyclic AMP. Cutting off the beta and gamma releases a lot of energy. A lot of energy that then can be used elsewhere in the cell. ATP provides energy for the making of the macromolecules, the DNA, the RNA, the proteins. Not only making the individual nucleotides for DNA and RNA or the individual amino acids, but the linking together of the DNA, the RNA into, long chain nucleotides, linking together of the amino acids to make polypeptides. Movement of these molecules, of these subunits around the cell takes energy. Moving things across membranes takes energy. Using the transport proteins to move them from one organelle to the next takes energy. And as I said at the beginning of this lecture, and I said in the previous lectures, all life on this planet seems to have evolved to utilize ATP as primary source of stored chemical energy. When it comes to, catabolic to anabolic uses. But we have to keep in mind that not only is there ATP, but there is GTP, UTP, CTP, TTP. All of those are present, but for whatever reason, ATP seems to be the one of choice. Now, there are certain reactions where some of these other triphosphates will be utilized. Remember, intracellular molecular cell biology talking receptors, G protein couples receptors. There's our proteins that utilize G triphosphate, not A triphosphate, as the energy source. The hydrolysis of that third phosphate of GTP is what is used to kickstart the reactions. What's interesting, though, is since we don't have substrate level phosphorylation, since we don't have, sorry, let me rewind, to make the triphosphates of CTP, GTP, TTP, UTP, we do not have oxidative phosphorylation that's going to be pumping those out. Okay, so what we need to do is utilize what's referred to as a ping-pong kinase event. ATP hydrolyzed to a diphosphate. The enzyme that does this takes that phosphate group and then will substrate level phosphorylate CDP, GDP, TDP, UDP, taking the diphosphate, turning it into the triphosphate. So instead of immediately pulling off the phosphate group off of, say, like PEP and doing a substrate level to C diphosphate instead of A diphosphate, like we will be talking about with glycolysis, and we've talked about a few slides ago, this ping pong steps have to take place where A triphosphate is the source of the inorganic phosphate, which an enzyme will pluck off of A triphosphate and then deposit it on the diphosphate. Here they say NDP or DNDP, N being any diphosphate that's not A. C, T, U, G. So you'll have things like the adenylate cyclase. Some reason your cells are under, demand, and your A triphosphate levels are diminishing, your A diphosphate levels are increasing, and your mitochondria just can't keep up, for whatever reason. You'll have things like adenylate kinases that will take inorganic phosphates and add them directly to A diphosphate to make A triphosphate. This is an enzymatic reaction because it's a kinase. So it's what they refer to as substrate level phosphorylation. And while it is quick, it is not as fast and is not as efficient as what we're going to see and what you probably talk about in other courses with oxidative phosphorylation and the ATP synthase. The creatine kinase uses the phosphocreatine, as I said, and takes that, strips off the phosphate, substrate level on A diphosphate to get the ATP back up again.